In our last video, we talked about how we can use secondary structural elements to come together to make an overall structure or overall fold of a protein. We call that the tertiary structure. One thing we didn't emphasize too much in that video was side chain interactions. This is really going to be the emphasis for both tertiary and quaternary structure, and I just want to touch on that briefly before we move on. Here's the structures of myoglobin and hemoglobin that we had on the previous page. We had a bunch of notes on there. I won't reproduce those here. However, consider when we have our groups coming off of secondary structures, when they project outward from alpha helices or from beta sheets, they're primed to interact with each other. We said in the last video that they tend to interact very tightly, or at least they're packed very tightly. And I want you to start thinking about what types of interactions might you have between different amino acids. For instance, if a leucine and a valine were right next to each other, what kind of interaction would they have? What if it was a glutamate and an arginine? What if it was a serine and an asparagine? What kind of interactions are possible between side chains? I'm talking about things that we've covered before in this class. We're going to revisit that later in this video. So let's go back and talk about the last and highest level of protein structure that we're interested in. This is quaternary structure. And what that is going to be the number and arrangement of multiple peptide or protein chains. Notice we've built up from primary to secondary to tertiary and came up with an entire protein fold. So what's left? All we've done so far, though, is fold up a single chain, one amino terminus connected all the way to the other end where there's the C terminus. However, that's not always the case. Sometimes you have multiple chains, each one having an amino and a carboxy terminus. They fold up and they then stick to each other. Let's take a look at how this works on the next page. Now, remember, on the previous page, we said that myoglobin and hemoglobin are globular water-soluble proteins and that they look a little bit like eggs. Well, here's what I didn't tell you. Although those eggs look a lot similar to each other, myoglobin only stays as one chain and has one egg-shaped structure. That's not true of hemoglobin, though. Hemoglobin, which we have shown here, doesn't stay as just a single egg-shaped tertiary structure. In fact, what we have is 4 egg-shaped tertiary structures that come together and stick to each other and form a much larger complex. Now, I've shown this both in the real structure and in a schematic diagram to the right. Let's take a look at the real structure of hemoglobin. One of the egg-shaped myoglobin looking like structures is found right here, and an identical one, you can see how they're both the same color, at least in mine, is found right there. Then there's another one here in gray, and that's identical to this gray one. So what can we say about hemoglobin's quaternary structure? Remember, quaternary is the number and arrangement of multiple peptide chains. Well, it turns out we have 4 total. 2 are identical to each other, and 2 others are identical to each other. And at this point, I would like to give you 2 different nomenclatures for quaternary structure. The first one is basically a word description of this. First of all, we're going to call hemoglobin a tetramer. And what that means is 4 subunits. Tetra means 4. Mer is a suffix we use for subunit, or really it means chain. Now, there's lots of other suffixes and prefixes we can use, and we'll practice that at the bottom of the page. But this is a tetramer, and that means 4 subunits. We put in front of that hetero. Hetero means different. And what I mean by that is that we have two of the white subunits that are the same as each other and two that are different. And that means not all of the subunits are the same. Maybe you can remember that we had hemoglobin A and B in our alignment page. That's the two different ones that form these different structures. They're very similar to each other, but they're not identical. If they were not identical, if one of them or more is different than the others, we have to call that a hetero whatever, heterotetramer in this case. Now, it could be that we have two of each type, or it could be we have three of one type and one of the other. Those would both be heterotetramers. So that's the first thing we want to use for nomenclature. The other nomenclature is going to be, believe it or not, Greek letters. We're going to use alpha and beta again. Now, this has nothing to do with alpha carbons, and it has nothing to do with alpha helices or beta sheets. This, again, is just the first and second type of structure we have. In this case, we would call this an alpha 2 beta 2 heterotetramer. That's the second type of nomenclature we use. That's two of one type, two of another type. So that's it. That's how we use quaternary structure nomenclature, and let's practice that. Go to the bottom of the page here. Here we have three subunits. I've made them all the same color over here on the right, at least. That means they're all the same. What would be the two different types of nomenclature that you would use for this type of protein? Pause for a moment and fill that in. Okay, I hope you had a chance to try that out. It's not that hard, despite the fact that I made them different colors over here. I just did that so you could see there are three different ones. I'm trying to indicate by the same white structure over here that they are all the same. We would refer to this as a homotrimer. Homo for the same, tri for three, mer for chains or subunits. And the second type of nomenclature that we would say is alpha 3. There are three subunits, but they're all the same, so we only have alpha in this case. Now, we're going to practice this nomenclature in the homework. It's pretty easy, but you can practice it over and over again until you get really good at this.